talk a bit now about kinship testing in forensic medicine, okay? And majority of kinship testing cases which we examine in our practice concern paternity testing, okay? So, which is the examination which has the higher demand in society because of many reasons, okay? So, so let's start with the general with the paternity testing examinations in forensic medicine. Okay, so there are several types of biological examination available for legal paternity testing. First of all, one can perform anthropological examination. Okay, there is a phenomenon which is very well known to all the people in the world, even to children, that children tend to be similar to their parents. Yes, they can share many external features, morphological features with the parents. Yes, just I don't know the color of eye. Pardon. What's going on? Okay, I have muted all the students now.
Dzień dobry, ja przed chwilą dołączyłem, Jacek Sobczak, miałem mały problem, żeby znaleźć link.
Okej, zaraz pana zaznaczę. Okay, and so in order to check whether it is possible that the man is a child's biological father, we can perform anthropological examination. We can compare these morphological features between a child, a mother, and a putative father. Okay, this type of examination is performed in Poland by some departments of anthropology of the universities or the Polish Academy of Sciences. It's based on comparison of approximately 180 morphological features. Yeah, just like the color of eyes, the shape of nose, the shape of ears, and many others. In order to perform this examination, a child must be at least 3 years old, so that all morphological features are developable enough. Okay, because when the children are born, they are usually not similar to anyone. Yeah, they are. I have 2 kids, and they were not similar to me after they were born. Yeah, so this is just one of the things we can use. However, this type of examination rarely allows us to prove or exclude paternity. Okay, because of the problem that these morphological features have very complex inheritance pattern. Yeah, there are many genes involved in the development of these features. Yeah, there is also some impact of the environment. Okay, so very often. Yeah, so we cannot. So it's very rarely possible to use the examination to prove that it's practically impossible to prove that somebody is the father, and it's very rarely possible to exclude paternity. Okay, the case which I remember that we could use, for example, anthropological examination, but the court ordered DNA testing, which is quite costly, yeah. But the court wanted DNA, and we were thinking, oh wow, why didn't they ask anthropologists to prepare the expertise? Okay, I remember there was a Polish woman who accused of a paternity a Swedish man. Okay, and this Swedish man came to Poland, to Gdańsk, to our department for collection of biological samples for DNA examination. And this man was just like a typical, how we can say, Nordic appearance. Yeah, just very fair hair, blue eyes, and so on. Okay, whereas the child had black skin. Yeah, so actually for me, it was, yeah, of course we showed in the DNA that he was not the father, but for me it would be much faster and cheaper to ask anthropologists and who would say, no, this man is not the father of the child. Okay, Another type of examination which can be performed, especially by men who want to prove that they are not fathers of the children, is to show that the man cannot have the children because he's infertile, because he cannot. So now let's imagine. Just one remark. If you say something, I'm just marking that you are active. Okay, so it matters a bit when I give you some small extra points, which may matter when I give you the final grade after the course. Okay, let's imagine such a situation that that there is a man in the court who claims this is not my child. Okay, I cannot have any children. I am infertile. Please look at results of examination of my semen. There are no sperms in my semen at all. Okay, so I was tested 3 times. Each time I was shown not to possess any sperms in my semen. Okay, so I'm infertile. I cannot. This is not my child. I cannot have any children. Okay, and now my question to you is, what do you think is the main drawback of such evidence? Yeah, how can you show that this evidence does not matter in this? Okay, can you write something on chat, or I don't know, or just say it loudly? What do you think? Of course, it's just that you do not have to know it. Yeah, it's just a guessing game. Okay, let me read the chat. Yeah, it's possible that he's not the father, but you are claiming that that he's not the father. However, from my point of view, I think that it is possible that he is the father. Why is it possible that he is the father? Anyway, why am I not sure? Yes. Yeah, exactly. Yeah. So who is speaking? Victor? Okay, Victor, and we can't tell if the results were different more than, okay, yes, Martina, okay, okay, yes, you are right. We cannot look at this results very often. We cannot look at this results retrospectively. Yeah, we know right now that he's infertile, but we do not know how it was, let's say, 9 months ago, as Martina suggested. Okay, so this is the main problem of such evidence. Okay, another type of evidence which may be used in legal paternity testing is based on comparison of a date of intercourse between a mother and a putative father with the degree of a newborn's development. Okay, it's known how long a normal pregnancy lasts. Okay. And based on the documentation from the birth, from the pregnancy, we can compute so-called conception period, when the child must have been conceived. Okay, and when we know the exact date of the intercourse between the couple, yes, and we have computed the conception period, we can compare these dates, okay? And sometimes it's possible to exclude paternity because the man does not match the date, okay? Actually, but this type of examination is useful quite rarely, because of the problems with the history of the couple, because one of the people claims, oh, we have met only once, and the other person say, oh, we have met several times. Okay, the other says, one of them says that they used contraceptives. The other says, the other person said, no, we did not use any contraceptives. Yeah, okay. So this is just these two people, the mother and the putative father, very often remember their relationship in a totally different way. Okay, and even if they agree on the date of intercourse and the date matches the conception period computed by us, okay, we cannot prove that he's the father, because it is still possible that the woman had intercourse with another man. Okay, we can use this evidence only to exclude paternity when the two people agree on the date of intercourse, which is totally different from the conception period. Yeah, so this is the only possibility. Okay, and another type of examination which is performed routinely at our department is the genetic examination, which is, and there are two types of research examination. The one is based on serology, and the other is based on DNA analysis. Okay. Okay, so now I'm going to tell you a bit about the application of serology in forensic medicine, just a brief historical background, okay? In 1901, this man, Karl Landsteiner, described the first human polymorphism, which is an ABO system, which is very well known to you, okay? The blood type ABO. He was given a Nobel Prize for this discovery 29 years later, okay? In 1910, mechanisms of blood group inheritance and basis of serological paternity exclusion were described, which and there were two researchers responsible for this discovery, Emil von Durgen and Ludwig Hirschfeld. The second one is the Polish serologist. Okay, so they two cooperated and described it. Okay, in 1916. For the first time, serology was used in a criminal case, and in 1924, for the first time, serological examination was used in a legal paternity testing worldwide. Okay, and in 1 year later, in 1925, for the first time in Poland, for the third time in the world, serological examination was used in a legal paternity testing case. It was done in Krakow, a city in southern Poland. Okay. Generally, you know what the blood consists of. And actually, all elements of blood contain proteins, which may show variations, polymorphisms between different human individuals. Okay, so we know that we have red blood cells, okay, erythrocytes, and they have different antigens present on the surface of these cells. Okay. They are on cell membranes, okay? They are ABO, MN, rhesus, KEL, DAFI, KIT, okay? They are different antigens, they are different proteins with different epitopes, okay? So they are different, okay? Then we have also proteins, enzymes inside red blood cells, okay? This is this third group, okay? And they can have different isoforms among different individuals, like acid phosphatase, phosphoglucomatase 1, esterase D, glyoxylase, and so on. So there are different enzymes found in red blood cells, inside red blood cells, which show variation between different humans, okay? And also we have proteins in the serum, okay? In the liquid between the intercellular liquid, okay? And so this is this list of this markers. Okay, so red blood cell antigens are detected mainly by agglutination, yeah, detected and differentiated by agglutination, whereas red blood cell isoenzymes and serum proteins are differentiated mainly with the use of electrophoresis. Okay, and sometimes also isoelectrofocusing is used to differentiate some subtypes of different red blood cell. Of different subtypes of different alleles. Okay, and then we have another group of extremely polymorphic proteins. They are called human leukocyte antigens, HLA, which are located on white blood cells, and they are divided to class 2 antigens and class 1 and class 2 antigens. Okay. And they are serologically differentiated with use of microtoxicity assay. Okay, so some brief information about some markers which I'm talking about. Red blood cell antigens like MN, rhesus, KEL, DAFI, KIT, and ABO. Okay, starting with the ABO system, which I guess is quite well known to you. Most of the people in developed countries know the ABO system and rhesus, antigen D presence. Okay, so this is the technical use of agglutination, and so first of all we detect antigens produced by a certain person. Okay, this is done by with the use of sera, some proper sera. Yes, so we add some serum to the red blood cells, and we check whether they agglutinate. And the other, so this is the first stage, and the second stage is always to confirm the result by detection of isoagglutinins. Isoagglutinins are natural antibodies produced by a person against antigens which are absent in the blood of the tested person. For example, my blood type is O. So in my blood, in my serum, there are antibodies, anti-A and anti-B. Okay. So first of all, my red blood cells will not be agglutinated with the use of serum anti-A and anti-B, but my serum will agglutinate red blood cells of type A and B, so which will confirm that my blood type is O. Okay. So also we use agglutinins from other organisms, mainly for plants, in order to differentiate antigen A subtypes. Okay, so here you can see some plant species which are used for this, to obtain these agglutinins. Okay, so so this is actually shown on the table what I have already told to you. Okay, that we have different red blood cell types with different antigens. Okay, present on the surface of the cell membrane. Okay, and there are different antibodies present in the serum, in plasma. Okay, let us go further. And here is just a result of agglutination. Yeah, so the anti-A serum agglutinate red blood cells of type A. So this is how we can see. So here, this is a result with no agglutination, and this is a positive result with agglutination. Okay, so here is just a brief information about genetics of the ABO system. So there are two main genes involved in the development of the system. Okay, so I would like to know this. I would like you to know this because it's actually quite important to understand the genetics of this marker. Okay, we have a gene encoding a fucosyltransferase. This is called H locus. Okay, which produces antigen H. Okay, and so generally all the people, majority of the people in the world produce antigen H. Okay, so they have antigen H. And this antigen H may become antigen A or B after addition of sugar by glycosyltransferase produced by the ABO locus. Okay, so when the sugar is N-acetylgalactosamine, then we will obtain antigen A. And when this sugar is galactose, then the resulting antigen is called antigen B. And when there is no addition of the sugar, only antigen H is left. So it's actually the O phenotype, which is a function of the ABO gene. Okay. So here you can see inheritance pattern of the ABO system. A1 allele is dominant over A2 allele, and this one is dominant over O, and also B is dominant over O. Okay. And there is something which I would like you to know, because there are some rare variants which I would like you to understand, and because one of the variants which is quite unusual is that it's called cis-AB. It's very rare, but it happens that some people have AB type, and actually majority of the people in the world, when they are at AB, it means that they have A allele on one chromosome and B allele on another chromosome. Okay, so they transfer to their children either A or B. Okay, but there are some people who are very rare, but they are known, which are called cis-AB. So it's possible that these people have AB already on one chromosome, and another chromosome can have anything actually. So this is just the standard situation. Okay, a couple AB and O, they can produce only children with A type or B type. Okay, because A is given to this child and B is given to this child, and O is given to this child and O is given to this child. But but there is something which is called cis-AB, that this person, let's assume that this person is cis-AB. It means that this person has, let's say, AB on one chromosome and O on another chromosome. It's possible that this AB was given to this child and O from another chromosome was given to this child. So actually, you can see that it's possible for an AB person to conceive a child with O type and vice versa. Okay. In the past, when people were not aware of existence of such a cis-AB phenotype, it was not possible to say, oh, AB person cannot have O child. okay? All person cannot have AB child because it's not possible. But now we know that it's also possible because of the existence of this rare cis-AB variant, okay? And there is another rare variant, which is called Bombay phenotype, which is actually found mainly in some, with low frequency, but it's found in Southern Asia, mainly. So it's this Bombay phenotype is a result of mutation in the gene encoding the H antigen. So there is no expression of H antigen at all. So actually, in order to differentiate this person, so we can differentiate this person by using anti-H antibodies. Okay. Normally, we always use anti-H antibodies, which agglutinate also red blood cells of blood type O. Okay. But when the person doesn't have H antigen at all, because this person is Bombay, we will not see any agglutination after we use anti-H antibodies. Okay, so let's go further. You can see frequency of different blood types in Poland. You know actually that these frequencies are population specific. And these frequencies are also used for statistical evaluation of the evidence of paternity in paternity testing cases. Okay, and this ABO system in Poland gives us 19% chance, because this parameter is population specific, okay, gives us 19% chance that the man who is not the father will be excluded from paternity, okay? So this parameter is called exclusion chance, okay? So 19% chance that the man who is not the father will be excluded, okay? It means that 81% of men who are not fathers will not be excluded because they simply match the child by random, okay? So actually, vast majority of men who are not fathers, after examination of only ABO, will not be excluded from paternity. Okay, okay, and the exclusion of paternity of men who is not the child's biological father is possible when the child has a dominant allele, which is absent in his mother and the putative father, okay? So you know what the dominant allele is, okay? A is dominant over O and B is dominant over O. And when the putative father has AB type, while the child has O type and vice versa. The second sentence is not valid anymore. I just showed you, I'm just showing it to you in a historical context, okay? Because you know about the existence of the cis-AB phenotype, so the second sentence does not allow us to exclude paternity, okay? Another example of another red blood cell system, this is MNSS system, which consists of two linked loci located on chromosome 4. So this MN system gives us 18% chance that the man who is not the father will be excluded from paternity. And together with the SS system, they gave us in total 32% exclusion chance. Okay, so let's go further. Another marker, rhesus antigens, okay? And there are two genes involved in the development of this rhesus protein, this RH protein. The first gene is RHD gene, which encodes RHD protein with D antigen. Actually, so many of you know about that you are Rh positive or Rh negative. So what does it mean? If you are, I am Rh positive, okay? So it means that I produce antigen D, okay? Which is antigen D, which is located on RHD protein encoded by RHD gene, okay? So, and Rh people who are Rh negative actually doesn't have the D antigen, okay? So we call them rhesus negative. They are rhesus negative. It means that they do not have the antigen D, capital D, okay? If we mark a person with a small d, because you are going to play a bit as forensic experts, I'm going to give you later some cases which you are going to work with, and you are going to tell me your opinions, okay? So this will be also some results of serological examination, okay? So if you see a small d, it is not an allele, okay? It is not a variant, it is not a recessive allele. It means actually that this person doesn't have the capital D antigen, okay? So this is just what you will see. And of course, inheritance pattern is that capital D is dominant over small d, okay? So the presence of the antigen T is dominant over lack of the antigen T, okay? And another gene involved in the development of this rhesus protein is RHC gene, which encodes RHC protein. and they are co-dominant antigens, capital C and small c. So remember, they are co-dominant, okay? They are co-dominant, even though there is a capital letter and a small letter. No, it doesn't mean that this is dominant only. And there are also another group of co-dominant antigens, capital E and capital E and small e. Okay, so they are also dominant. We have a serum against capital E and we have serum against small e. So they are different antigens. Okay. We have also a rare variant of capital C antigen, which is called capital CW, which is detected with anti-CW antibodies. Okay, so just to tell you that there is some such a variety, which is routinely detected also in our examination. Okay, and this rhesus system gives us 31% chance that the man who is not the father will be excluded from paternity. Okay, and another system of red blood cell antigens. This is Kel-Solano system. There are two co-dominant alleles, capital K, known as Kel, and small k called Solano. And actually, in our practice, we only type the capital K antigen with proper serum. So we divide people into people who have this capital K, this Kel antigen, and people who do not have this Kel antigen. Okay, so let's go further. It's not so. Actually, because of the distribution of these people who have this Kel antigen, okay, look, only approximately less than 9% of people in Poland have have this Kel antigen, okay. So actually, because of this distribution of the frequency of these alleles in Poland, this marker gives us only 4% chance that the man who is not the father will be excluded from paternity. Actually, so it is not very useful in practice. Okay, there are some other red blood cell blood types, so which are listed here in the table, but it's not so important. Let's go further. Some examples of the serum proteins. Okay, let's go further. Okay, an example: haptoglobin, which has two main alleles, HP1 and HP2.
Excuse me.
Yes.
Can I have a question before you move to another topic?
Yes,
about
sure.
the HB0 groups.
Yes.
Could you explain the difference between A1 and A2?
Yes,
this is something I've never heard before.
Actually, the A1 is a result of strong expression of the antigen. So A2 it means that this person has a lower density of the antigens on the red blood cell membrane. Okay. So this is not just that they are different epitopes or something like this. No, it's actually the result of lower expression. So the density of the antigens is lower. Okay.
Okay. Thank you.
Let us go further. So, and this allele 1 of the haptoglobin may be differentiated into subtypes. Subtype F, it means fast. Subtype S means slow by isoelectrophoresis. Okay, so normally we perform electrophoresis to detect these two main alleles, and also isoelectrofocusing can differentiate two subtypes of HP1 alleles. Okay, F and S. Okay, so exclusion chance for this marker is 18%. Some other examples, yes. So with examples of electrophoresis, another marker. So it's not so, how to say, we do not have to go into details so much because it's just very general information, and what you have to remember is the technique electrophoresis and isoelectrofocusing as a method which allows us to differentiate some subtypes of these markers. Okay, and some red blood cell isoenzymes found inside red blood cells. An example of electrophoresis of the acid phosphatase system, which is differentiated by electrophoresis in a starch gel. Yeah, you can see in Poland we have 3 main alleles and 6 main phenotypes, which look like this after the electrophoresis. So after we see such a result, we know that this person is A or BA or B or CA or CB or C. Okay, the order of the letter is different than in the alphabet in order to make it different from the ABO system, from the AB phenotype in the ABO system. So people decided, let's call it BA, so it looks a bit different from AB in the ABO system. Okay, so this is why they call it BA. Okay, let's go further. And this marker gives us 26% chance that the man who is not the father will be excluded from paternity, okay? And some other markers, the same story. Electrophoresis as the main technique, okay? And iso-electrofocusing as a method which allows us to differentiate some subtypes of the phosphoglucomatase 1 system, okay? Let us go further. Esterase D, the same story. Glycosylase 1, the same story. And let us go further. Okay. So here you can see the exclusion chance values presented from different serological markers. Okay, some markers are more useful for paternity testing, some markers are less useful. And for example, let's see, the cal is not very useful yet. The rhesus is quite useful, yeah. But remember, we never test in our practice. We never test only one serological marker. We always test more markers. But look, even if we test as many as 18 markers, yeah, a lot of work, a lot of time, a lot of reagents, yeah, so separate electrophoresis of another marker, and a lot of work, really. So there's only 97% chance that the man who is not the father will be excluded from paternity, okay? So this is just the main problem, that we cannot, yes, we are quite lucky that if somebody is not the father, we may be quite lucky that we will show that he's not the father, but if he is the father, we cannot prove that he is the father. Yeah, maybe he matches because he was among 3% of men who are not excluded after this examination. Okay, so we can also include examination of human leukocyte antigens, which are highly polymorphic. Okay, which are serologically we are tested with the use of microcytotoxicity assay, also known as Terasaki's test or NIH test. NIH stands for National Institute of Health. Is an institute where this, in the US, where this test was developed, okay? So how this test works? So we have our cells, yeah, we have our leukocytes, and we add to our leukocytes, we add serum, we add also complement, okay? We also add dye, which is eosine. If the antibodies from the serum recognize antigens present on the cell membranes of the leukocytes, the cell membrane is damaged by the complement, and the eosine dye enters the cells, and the cells are stained. If there is no recognition of the cells of the antigens by the antibodies from the serum, the cell membrane is not damaged by the complement, and the eosine stays outside the cells. Okay, so here is an example of the positive result. Under the microscope, you can see that the cells are stained with the dye, with the eosine. So we know that the antigen that we were looking for with our serum is present in this individual. Okay, so this person produces this antigen. Okay. And here is an example of a negative result. Okay, the cell membrane was not damaged by the complement, so it means that there was no that the person does not have the antigen for which the serum was used. Okay, so this is, you can see where that these results look totally different under the microscope. Okay, and of course, currently, especially for transplantation purposes, the HLA typing is performed with the use of molecular techniques, with the PCR, with the use of sequence-specific primers, of subsequent hybridization of sequence-specific oligonucleotide probes. So these are two techniques which are used the most commonly. And they can also be used for in some cases for paternity testing. Okay, so let's look at this table of values of parameter of exclusion chance after inclusion of the HLA, and we can see that if we include 68 antigens, the cumulative exclusion chance increases to 99.41%. Okay, if we include all known antigens for loci A, B, and C, this value is even higher. But there is still there's a lot of work, a lot of costs. Yeah, really extremely laborious, and there is still a very small percentage of men, but there is, who are not fathers, but will not be excluded from paternity. Okay, so since it's so laborious, it's so costly. In order the standard serological examination includes approximately 10 serological markers. So this examination has some economical justification, okay? So these 10 markers give us approximately 90% chance that the man who is not the father will be excluded from paternity. It means that if the man doesn't match the child after serological examination, we write in the conclusion that he is not the father. But if the man matches the child, we do not suggest that he is likely the father. It's possible that he's the father. No, because it's still possible that he's not the father, but he had bad luck and was among 10% of men who are not excluded after standard serological examination. Okay, so just remember that if the man matches the child in serological examination, we are not allowed to suggest that he's the father. It's likely that he's the father. No, we write that what we write is that paternity cannot be excluded. Okay, so this is just the safe sentence which does not tell us anything actually about the possibility of paternity. Okay, okay. So now I'm going to tell you a bit about DNA examination in forensic medicine. Would you like to have a short break now, or shall we continue? What do you think?
We can do a short break.
5 minutes is okay?
Yes.
Yes.
Okay. Okay, so let's have 5-minute breaks, okay?
Hi, can I have a question? It's not a question about the class, but I just connected a couple of minutes later because I had problems with connection, and I just wanted to check if you had checked the attendance.
Okay.
My name is Miriam.
Yes, I have checked, and it's marked that you are absent, so now I'm marking that you have come. Okay. And you have joined us. Yes, Miriam, Lai.
Yeah, thank you.
You have joined us later. Okay.
Nie, ja też się spóźniłam.
And your name is?
Karolina Milcz.
Karolina Milcz. Yes. Okay. Mm. 24. Okay, so there's only one person absent in this. Yes, am I right? Zuzanna Krela, are you here with us? Okay, okay, everything is correct. Okay, welcome back. Okay, we are continuing. Okay, so now, as I told you, we are going to talk about the DNA examination in kinship testing. Okay, so just to brief you, remind me of, I guess you already know of what you already know quite well, is that the structure of the human genome, which consists of the nuclear DNA and mitochondrial DNA in the nucleus. This genome consists of 23 pairs of chromosomes, 22 pairs of autosomal autosomes, and 1 pair of sex chromosomes. Okay, so I'm sure you know it and remember it. Okay, so you know it's localized in the nucleus and also in the mitochondria. And how it is built, I'm sure you remember. So let's go further. So what is important about DNA regarding the kinship testing and generally forensic genetics is that DNA encodes genetic information. So actually how we look like, also some features of our behavior, yes. So, and sometimes also, okay, so some genetic disorders, predisposition to develop some complex disorders, okay. And the other thing which is quite important for us is that this information, which is encoded by DNA, may be precisely replicated. Yeah, replicated and transferred to daughter cells after cell division, and also is transferred to next generation after the as a result of inheritance. Yeah, just it's transferred to our children. Okay, so and this information, this transfer of the genetic information of these sequences of DNA is used in kinship testing. Yeah, it's transferred to next generation from parents to children. Okay, so generally when we look at the human genome, only 2% of the genome is covered by the coding sequences, by some sequences which encode some proteins, some RNA molecules, okay? So, and generally, so whenever you talk about medical genetics, human medical genetics, you think of the coding sequence of the genes involved in the development of some monogenic disorders, some predisposition to develop some complex disorders, some genes involved in the development of cancer. Yeah, so they are mainly genes. Okay. So however, forensic geneticists are interested especially, but not only, are interested especially to non-coding sequences. The vast majority of forensic genetic examination nowadays in the world is examination of the sequences in the non-coding DNA. Okay, and these non-coding sequences comprise 98% of our genome. And among these non-coding sequences, majority are repetitive sequences. Okay, which are sequences which are repeated because of different reasons, okay? This can be, for example, transposon-derived repeats or simple sequence repeats, okay? As some example, this transposon-derived repeats, which comprise 45% of our genome. An example which is maybe known to you is the allo elements, okay? Which is an example of the short interspersed nuclear element, okay? And this allo element has a length of approximately 300 base pairs. And there are over 1 million copies of this allo element in the human genome. Almost 11% of our genome, these are these allo elements, okay? And however, and less than 0.5% of this allo element are polymorphic. What does it mean polymorphic? That some people in this fragment of the chromosome has insertion of the allo element, and some other people doesn't have. It's insertion of 300 base pairs, and so and some people do not have okay? Or so it is just possible to check whether the person is just, so it's very basic binary, it's very basic binary polymorphism in the human genome, just to check whether the person has alu element in this region or it doesn't have this alu element. Okay, so because the fragment will be longer or shorter because there is this fragment is absent in the genome. Okay, so it's, and sometimes this alu element is used for some anthropological examination of humans, yeah, just I don't know, to trace some human migrations, to relationship between different humans, okay? So, and it's also possible to use this alu element in forensic genetics, okay? However, what forensic geneticists analyze the most often are simple sequence repeats, which comprise 3% of our genome. And they are divided into mini satellites and microsatellites, okay? So this division actually has some technical background because initially DNA polymorphism was analyzed with the use of hybridization with multi-locus and single locus molecular probes. And this molecular probe, this analysis with the hybridization, by hybridization with molecular probes, was able to differentiate or to detect polymorphism of only mini satellites. Okay, so this is what, and later PCR technique was developed, which allowed genotyping of both mini satellites and microsatellites. Okay, and this technical division actually results from the size of a repetitive unit. If this unit is very short, yeah, up to 6 base pairs, this is a microsatellite, also known as short tandem repeat, STR marker. Okay, just remember microsatellites and STRs, these are synonyms. Okay, and also PCR was used to examine the variation of many satellites. Okay, so let's go further. And this is just a table which shows that in our genome, the most common STRs are STRs with D nucleotide repetitive units. Okay, so they are the most common. Okay, and when you think of DNA examination in forensic medicine, there are different applications which you can think of. Okay, most of you think of forensic cases. And this forensic examination consists in matching, comparing DNA profiles, yeah, whether they match. We have a suspect or suspects, or sometimes we have a victim, a profile of the victim, and we compare DNA profile obtained from the evidence material. This evidence material, maybe, I don't know, some blood stain, I don't know, some swab from a body of a victim or swab from a body of the suspect, yeah, and we compare these profiles whether they match, yeah, and sometimes, and even quite often, it's possible to obtain a mixed profile of several individuals, of at least 2 individuals. So our task is to assess statistically, then in this mixed profile, the suspect's profile is also present. Okay, so this is just what we are going to do. Okay, another application is the kinship testing, and majority of kinship testing cases are paternity testing cases. Okay, and another application is concerns historical investigations. Okay, some quite interesting historical questions were answered thanks to genetics, forensic genetics. Yeah, and I have prepared such a presentation with quite interesting cases from our practice and also from the world of literature, just also including some historical questions which were answered thanks to forensic genetics. Okay, another type of application is the concerns investigation of the missing persons. Okay, and identification of cadavers. Okay, at our Department of Forensic Medicine, we have autopsies. Very often, we have autopsies of cadavers with unknown personality. Okay, so we have some bodies and brought to our department, yet the identity of the person is not known, or there's some suspicion who this person may be, and our task is to confirm this suspicion or just to try to identify this person. So routinely and the most often is performed by comparison to DNA profile of the family members of the putative family members. Okay, so this is what is used the most often. Yeah, another application is the identification of victims of mass disasters. Luckily, our department. So there are two types of mass disasters. Mass disasters are classified as open mass disasters. Open mass disasters are disasters in which we do not know the exact number of the victims, and we also do not know very often personality of the victims. Yeah? Can you tell me an example of an open mass disaster? Pardon? Open mass disaster, yes. Tsunami, yeah, you are right. David Torem, yes, you are right. Okay, very good idea, yeah. Airplane crash. Do you think that airplane crash is an open mass disaster? Yeah. Actually, what is avalanche? I do not know the word. Avalanche or Kacper?
Ten
Okay.
śnieżny jak śnieg zasypie ludzi. Też mi nagle polskie słowo uciekło.
Lawina.
Tak, lawina.
Lawina. Też chyba mi się wydaje. I think that when the snow goes down from the mountains, yeah, there may be quite many people that we don't know. We do not know how many people died and their personalities. Yeah, but it's it's not so easy because maybe there can be only several victims and we know who was on the, I don't know, skiing right now. So, and we know that they just died. Okay, so they are missing. Yeah, so this is actually, it's not so this assignment is not so strict. Okay, in this case of this avalanche, or however it is pronounced. Okay, but airplane crash, no, I think when we have an airplane crash, we know the names of the victims. Yeah, and we know the number of the victims. So actually, this is a very good example of the closed mass disaster. Okay, so this is just the other type of mass disaster. Okay, luckily, our department doesn't have much experience with DNA examination of the mass disasters. We have only tested some cases in which there were only several victims. So I remember some cases. I don't know that there was like two teenage boys who were driving a car, who were going by car, and they had a car accident, and the car was smashed so much that it was totally impossible to identify the two victims. Yeah, one of them was a driver and the other was a passenger, and so it was actually the only way to identify the bodies who was with the use of genetics. Okay, so I remember, I don't know, is there such a word like helicopter in English? Do you know the word? Am I right? Is there such a word, helicopter? Or is it my invention? Yes, okay. So I remember a helicopter crash. There were like three victims who died. So we were identifying them. One of the passengers was the husband of a Polish parliamentary member. So it was quite announced. There was a lot of information about this crash in the media. Okay, and I remember also like a crash that there was several people who died. There were sailors who were sailing the yacht in the Masurian lakes, and they touched the energy line. Yeah, so the yacht was burned totally with the bodies. So our task was to identify the sailors from the yacht. Okay, so this is our small experience with the mass disaster. That there were luckily not so many victims. Okay, and DNA, another application concerns DNA, military DNA dog tags. You think of this pieces of, when you think of military dog tag, you think of this pieces of metal worn by soldiers, okay? But also DNA profiles are unique and they are also determined in case of soldiers. So they, in case it's necessary, they are also easily identified, okay? And another application is the development and establishment and maintenance of databases of DNA profiles of convicted felons, okay? So there is, because different studies show that if a person was in a prison, it's quite likely that after this person leaves the prison, will commit another crime. Okay, so actually, I know also the story that the person who entered the prison even became worse. Yeah, I remember this terrorist attacks in Western Europe. Yeah, that I have read some stories that this person was punished and was spent, I don't know, some time in a prison, and in this prison this person became radicalized. Yeah, so actually the socialization and success of this person was totally negative. Yeah, actually he became even worse in the prison than in comparison that what he was before he was punished. Okay, so actually this DNA databases with the profiles of these felons facilitates a lot the work of police, prosecutors, very often to identify very quickly who committed the crime if the DNA profile is already in the database. Okay, and an example of such a database, which shows the highest usefulness in the world and the highest percentage of detection of profiles and in criminal investigations, is the British DNA database, because actually some human right defenders claim that this UK database violates some human rights, because if a person is tested and the DNA profile is is introduced into this, is put into this database, actually it will be left in this database. I'm probably forever. Yeah, because it was very difficult to be removed from this database. Even if you are shown that you are totally innocent, your DNA profile is in this database. Okay, so some people claim, oh, this is violation of the human rights. So if the person is innocent, why DNA profile should be actually stored in this database? Okay, actually, from my point of view, if I am innocent, actually I can publish my DNA profile, I don't know, online on my Facebook account, and you can just read it. But in some crazy countries with some strange regimes, or sort of, sometimes I think that maybe this information, it wouldn't be so good to present this information and know to everyone in the world. Yeah, so let's go. Okay, so generally, yes, so there is a high percentage of cases, forensic cases, solved accidentally in the UK, thanks to this large number of profiles present in the database of DNA profiles. Okay, let's go further. Okay, what type of biological examination may be used for extraction of DNA? Okay, generally we can use to extract DNA directly from blood. Okay, we can collect blood samples, and for DNA examination, the best anticoagulant which is preferred is EDTA, and another type of material which we encountered and we quite often in our practice are FTA cards. What are FTA cards? Actually, this is a type of paper which is covered with some special substances, yeah? And according to the producer, because this is a commercial name, FTA, according to the producer, when you put a drop of blood onto this paper, the cells are lysed, yeah? DNA is bound to this matrix, yeah, and is protected from degradation. And also, if there are any pathogens present in the blood, they are also inactivated. Okay, so this is a very good way of storing, of storage of high quality DNA at low temperature. And also, if you have to send your DNA sample from just from one laboratory to another, just at room temperature in an envelope, it's very convenient to use this FTA cards. Okay, so this is just you may is good to know about such a way. Okay, and another type of material which is encountered very often by us and is also collected very often by us are the buccal swabs. Okay, because it's let's say in case of serological, let's come back for a moment to the serological examination, the blood types. Okay, in case of blood types, when you have to perform blood typing for for paternity testing, we have to wait until the child is 6 months old, half a year old, so that all serological markers are developed well enough. But even if the baby is half a year old, we have to collect blood for serological examination, okay, to type the blood antigen. Okay, so we so we have to collect the blood. So we have to find a venule in the hand of this baby, yeah. And sometimes these babies have quite fat hands, yeah. So it's quite difficult for us to find this venule, yeah. So we try with one hand. If we are not successful, then we try with another hand. And if we are still not successful, then we collect the samples, the blood from the fingers, yeah. So there is a lot of crying of a baby, and the baby wants to run away, and we have to use force to collect this blood. For me, it's a bit cruel, actually, because it's not for any medical purpose. It's not for any, I don't know, medical treatment, medical diagnosis. No, it's only to test for paternity, yeah. But in case of DNA examination, first of all, we do not have to wait any time. We can collect the samples immediately after the birth, and we can do it in a non-invasive manner, totally non-invasive manner, in the form of buccal swabs. Okay, so these babies, even they don't know, a few weeks old, so they come to us with the mother, yeah, and we swap them, and they even sleep. they even do not wake up totally, yeah. So it's very good for us. So it's not invasive. It doesn't induce any stress, or at least sometimes there are children who get stressed also because of this, yeah, because of the white coat, okay, for example. But yes, so it's very convenient to us to collect the samples in the form of the buccal swabs. Moreover, DNA examination is also possible in prenatal. Okay. In the past, in order to, because sometimes it's important to know who the father of the child is before the child is born. Okay, and so in the past, this examination required some invasive sample collection procedures, yeah, like amniopunction, yeah. Actually, this procedure bears a small risk for the pregnancy, yeah, to lose the pregnancy, yeah? Very low, but there is still. But so it was actually. But even though, but right now we can test for paternity prenatally in a non-invasive manner by analysis of DNA, fetal DNA, which circulates in the maternal plasma. Okay, so we collect blood from the mother, yeah, and there is some DNA or fetal DNA circulated in the plasma. So it's possible also to test for paternity prenatally in a non-invasive manner for the pregnancy, okay? But in the past, I told you, sometimes it was so important to get to know who the father of the fetus was that the police, the court, yeah, decided, yeah, you have to do it right now. So that the doctors had to do it, even taking into account the very small risk of damage to the pregnancy. Okay. So what cases am I talking about? Do you have an idea? In which cases it was so important to get to know paternity of a fetus, even prenatally, yes, just and even so that invasive sample collection procedures were practiced?
Someone suspect genetic disease.
If there is a suspicion of a genetic disease, okay. Why to test for paternity? Isn't it better to just to check whether this fetus has a genetic disorder directly? Yeah. Why to test for paternity? Actually, there is a good answer on the chat. Yes, Victor, you are right. If there is a suspicion that the fetus, that the pregnancy is a result of a rape, okay, or another crime, that it be incest, yeah. So why is it important to know, to prove that the fetus, that the pregnancy is a result of a rape? Do you have any idea? Actually, it's specific to our country, to Poland. It's connected to the Polish law. Okay, there is some. Yes. Who was speaking? Victoria. Okay, yes. And also Carola. Okay, yes, you're right. Okay. Yes, exactly. Because according to the Polish law, there is still it is still not. It has not changed until now, and I'm not sure if maybe they will change it even in such cases. The abortion is possible in Poland when it's a result of a crime. Yeah, let it be rape or an incest. Yeah. So this is just, and sometimes it was important because the woman got pregnant and there was a question who the father of the fetus was, or whether it was a rapist or a husband. Yeah, and I know a case from our practice that we saved the pregnancy because we showed that the father of the fetus was the husband and not the rapist. Okay, let us go further. And another type of material which we analyze routinely in our practice are soft tissues. Because sometimes it's not possible to get the blood sample from the cadaver, yeah, because it's so bad that it's only possible to collect the soft tissues. Usually we get fragments of skeletal muscles, yeah, when it's, this tissue is still quite fresh and pink, yeah, and sometimes the body is decomposed so much or is mummified, or there's only some remains that we analyze DNA from hard tissues, from bones or from teeth, okay? And just, it's good to remember to you that the bones protect DNA very well, yeah? Remember from histology, there are some osteocytes which are inside this hard tissue, yeah? So this DNA inside the cells is quite protected by this hard tissue from degradation. And what is important to remember is that very often the age of the bone is less important to us. What is more important for the success of genotyping is the conditions in which this bone was stored, yeah? Because if this place was cool, yeah, and dry, this bone can be really very, very old and we still may be successful in genotyping. But if this place was warm and with high humidity, then even this bone will start decomposition, will start undergoing decomposition, and then the DNA will become degraded. Yeah. So just to remember, the conditions are much more important than the age of the bone. Okay. So the main problem is just to get this DNA out of this bone because it's hard. Yeah. So what we have to do is we use a mill. We have a special mill for milling the bones. Okay, so we cut a small piece of bone, yeah, with a saw, yeah, and so we have to clean it very, very, very well. Actually, I prefer older bones, which do not have any soft tissues, because sometimes I get this bone which is covered with some decomposing fragments of, I don't know, of muscles or whatever. So there's a lot of very bad smell, and also inside this there is this fat marrow, so bone marrow, so this also stinks terribly. And sometimes there are even worms inside this alive worms inside this marrow, bone marrow. Yeah, so we have to remove everything and a lot of really. So it's actually I prefer very old bones because they are the comfort of the work is much better. But anyway, we have to clean very well these bones and also to remove any impurities, also to remove any external DNA, exogenous DNA, which may be responsible for contamination. Yeah. So we put this clean, dry bone, which is treated with sodium hypochlorite, which is very strong oxidizer. Which is used, for example, in toilets. Yeah, I guess you know Domestos. Yeah, a trademark. Yeah, so Domestos is a product used in toilets and contains sodium hypochlorite, which is chemical which is used in forensic laboratories for because it kills germs. Yeah, it kills fungi, bacteria, viruses, but it also damages. Kills DNA. Okay, so in forensic genetics, we use it for cleaning any tools and surfaces and so on. And also, I put a piece of bone into this solution of sodium hypochlorite to kill any exogenous DNA of a person who was mainly touching this bone with any I don't know impure hands or whatever. Just so this is, we also use the UV light, which also modifies DNA in a way that it's not possible to amplify it. So yes, also the UV light is used so then we put this piece of bone into a plastic tube with a piece of metal, and we put it in this mill, and there is a very variable electromagnetic field applied to this tube. So this piece of metal starts moving very rapidly inside this tube with the bone. So this bone is crushed into the powder, and this process is so rapid, that it generates so much heat that our sample will be destroyed actually. This is why this milling process is performed in liquid nitrogen. So we pour liquid nitrogen into this mill, so everything, so it makes so that it protects this bone from heating and DNA, protects DNA from degradation during the milling process. Okay, so this is how we produce this powder from bone right now. In the past, when known mills were available in the market, this powder was produced in a way that we were using a saw, and we were cutting this bone with a saw, and there was some powder produced during the sewing process. Yeah, and so we were collecting this powder. A lot of work. I remember I had like a how do we call it? Some muscle aches. Yeah. So there was because of this hard work with just cutting this bone. So, but this is but right now we have this mills for milling the bones. Okay. So another source of material which is used by us quite routinely, source of DNA for forensic examination is archival material, which is this archival material may be paraffin, formalin preserved, paraffin embedded tissues, or histological slides. According to the Polish law, if pathomorphological examination is performed and paraffin embedded tissues are produced or pathomorphological slides are produced, they have to be stored by an institution for 20 years, okay? At least 20 years. If there are cytological slides produced, they have to be stored for at least 10 years, okay? So this type of material sometimes is possible to, that the only material, biological material, which is available for genetic testing, for example, for paternity testing, is this archival material. Yeah. And very often, in order to check, prove, to examine the cause of death of a person, a pathomorphological examination is performed at the departments of forensic medicine. Okay, so some fragments of tissues are collected from the cadaver, yeah, and they are preserved in formalin, they are embedded in paraffin. Histological slides are prepared, and they are, so pathomorphologist value is looking for the cause of death under the microscope. Okay, so it's also it's used in the practice quite often. Okay, so I told you that this material is stored for years in our department, and sometimes we have paternity testing towards a man who died. Okay, and we know that he had autopsy at our department. We go to our archive. We know that some tissue samples were collected from him. We go to our archive, we get the samples, and we check very quickly whether he is the father of the child. Okay, so this is one of the ways how to check paternity, whether if the putative father is dead. Okay, so my question to you is now, please give me some other examples. What else could we do if the putative father died? What else could we do? I told you one of the solutions, about one of the solutions. There are many options actually. Use your brains, not knowledge, but on your creativeness. Test his relatives. Very good idea. Yes, I agree with you. So it's actually very, very one of the most commonly used solutions is to test his relatives. Yeah, his parents, his children, if he had other children, his siblings. Yes. So it's actually further more distant relatives are not used because they have too few features in common with the putative father. But indeed, parents, children, siblings are used quite often to test for paternity if the putative father is dead. Okay, some other solutions. What else could we do? The putative father is dead, and you have the task to help the mother of the child to perform DNA test. Okay, we can test his relatives, but his relatives said, no, we will not take part in this examination. So these are civil proceedings, they are not criminal proceedings. So the people must agree to participate in the examination, okay? So they say, no, we will not take part in the examination. Or the putative father doesn't have any, didn't have any relatives, okay? So what else could we do? What would you suggest to the mother? What could she do?
Maybe somewhere left some of his hair, for example.
Mm-hmm. Yes, what kind of hair, what kind of, where, what?
On the brush somewhere in home.
Yeah, brush or comb, yeah, you are right, yeah. What else could this be? Some other personal belongings which may contain his DNA. Mm-hmm.
Toothbrush.
Toothbrush, yes, very well. You have some other ideas? Toothbrush or the hair comb, yeah, what else? Something which is also used by one person. More often by men. What am I talking about? Razor, yeah. Mm-hmm. David, yes. Razor, yeah, just the shaver, yeah. This is a, so yes, we sometimes have we get some personal belongings to check personality of the cadaver, yeah. So for example, we get a DNA, so we get, I don't know, like the shaver, yeah, just to check whether the profile from the shaver matches the cadaver from our autopsy, okay, so that we can prove identity. Yeah, but I can tell you that it happens very rarely that we have such material for examination. Why? Because first of all, because such personal belongings, which are used only by one person, are very often thrown away after the death. Yeah, they are not stored. Yeah, because they were personal. Yeah. And moreover, we have to be sure that this was the only person using it. Yeah. I remember even a case which may shock you a bit, that we had a cadaver from our department, at our autopsy room. Yeah. And we determined DNA profile of this cadaver, and the police sent us DNA profile from a toothbrush. And actually, what was surprising to me was that they found a mixed profile of two individuals on this toothbrush. Okay, and the dominating profile dominating the quantity. Okay, because the peaks were higher, was DNA actually matching the cadaver. Yeah. But it means, okay, something which is very personal, which is used only by one person, is a toothbrush, yeah? But actually we found, oh, there's DNA of two people, yeah, at least, yeah? Maybe somebody will just touched it, but I guess, but it's not possible because there was so much DNA of the other person. My explanation, which may be a bit shocking to you, is maybe that the toothbrush was so young, so new after the death, that they decided to use it because they didn't want to throw it away because it's so new, so that someone else started using it. But this is just maybe it's not maybe it was not. It is just an explanation which is not maybe not true. Okay, but so this is the other problem. Yeah. So we have never had a case of paternity testing in which we used such things like as a shaver or comb for paternity testing. Yeah, we had cases in which we used such belongings for identification of a cadaver's personality. Okay, so okay, some other options. What else could we do? The putative father is dead. What else could we do? His personal belongings, his relatives. He had autopsy at our department, and we have tissue samples collected from him. Yeah, so this is some other solutions. What else could we do? Where is his DNA in large quantity?
Extremely we can dig
large
out
quantity.
His grave.
Yes. So what can we do? Yes. What can we do then? This is a very good answer. Yeah. What else? What
if
can we do then?
We open up his grave, we can take as many as we want samples of his.
Yes, exactly. Elisaveta, yes, you are right. Am I right?
Yes.
Okay. Yes, exactly. So this is what is also performed sometimes for paternity testing. Yeah. We can exhume the body. Yeah. We can get the body from the grave, out of the grave, and we can collect the samples and put the body back to the grave. Okay. So this is also it's sometimes the only possibility to check this, to prove this in this way. But it's also another problem that is that I remember in cases in which the family didn't want to participate in the examination, and even didn't agree to exhume the body of their father. Yeah, so they were totally against an examination to check whether they have another brother for division of any possessions or whatever. Yeah. So this is just, but it's performed, and we have done quite many such examinations in which the body of the putative father was exhumed for paternity testing. Okay. So very good idea. What else could we do? Mm-hmm. Where can we find maybe some his tissues? I don't know, maybe some his cells, maybe even alive cells somewhere. He is dead, but maybe there's, or maybe some information in some, I don't know, some servers, on some servers, some computers, some information which may be used directly for paternity testing, or maybe some DNA, some tissues or some cells which maybe are even alive.
He was a criminal, then his records must be in base.
Perfect. Yeah, exactly. Maybe he was a criminal. Yeah. And Elisaveta, yeah, again, yes. Am I right? Yes.
Yes. Yes.
Okay. Mm-hmm. Yes, you are right. Maybe he was a criminal, yeah, and his DNA profile is already in the database of convicted convicted DNA profiles of convicted felons, yes. So, and this profile, which is stored in this police database, may be used directly also for paternity testing. Okay, so this is one of the way. And also maybe he had already participated once in his life in another paternity testing. Okay. And also the same profile which was used, which was obtained in the past, may be also used again for paternity testing towards another child. Okay, so this is, we can use DNA profile which is already stored somewhere, yeah, by in any document or the database. Okay, so this is a very good idea. Okay, some other examples where we can find maybe his tissues stored somewhere.
Maybe he donated some blood or another tissues to some somewhere.
Maybe if he donated blood, yeah, but the blood, how to say, there is just a short time where this when this blood is maybe used, yeah. So it's when he died, I don't know, 1 year ago, there will be no blood at all, yeah. Even if he donated his blood and there's a person who has his blood, this blood is exchanged after a few weeks, yeah. So, but the tissues, what kind of tissues are you thinking about?
Could we also check the body marrow?
Bone marrow, you mean?
Yeah, bone marrow. Because
Okay,
bone
yes,
marrow should last longer in someone else's body.
Yes, exactly. Adrian, yes, but I will ask that Elisaveta is active, yeah, and Adrian, okay. Yes, you are right. Actually, we can, if he was a bone marrow donor, we can get, we can find a recipient of donor, yeah, and sorry, the recipient of the bone marrow, yeah, and we can simply get a blood sample from this person because it's not so invasive to collect a blood sample from a person, yeah. If he was a kidney recipient, no, it just to get a biopsy of the kidney is quite invasive, yeah. But just to get a blood sample, it will not be very very invasive, yeah. Okay. So this is one of the possibilities. But I can tell you, we have never done such examination in our practice. It's just theoretical possibility. Okay. Probably maybe someone used this possibility in the world. I'm pretty sure it was done. What else could we do? Maybe there's some other place where his alive cells are stored. What place am I thinking about? Yeah. Sperm umbilical cord. Yes, you are right. Sperm bank, umbilical cord blood, spanks are. Yeah, you maybe you know that after the birth, sometimes the parents want the umbilical cord blood to be stored for longer, yeah, and they pay for it, yeah. So this is one of the options. Maybe we have said this blood sample stored in the bank, or maybe he was a sperm donor and there is his sperm stored in the bank. So it's also a possibility that we could use this, yeah. So David, you are right fully. Victor and Carola, okay, and there's one new message. Organ donor after death, that he was an organ donor after death, yeah. But is there a problem with the biopsy, yeah? So this is just, it is possible, yeah, we're right, but maybe he was given, I don't know, he gave his I don't know, even heart, yeah, but how to get this heart sample for paternity testing? We would have to wait until the recipient is dead, I guess. So it's too complex to me, yeah. But I would like that you have suggested it, a very good idea. But there is another place where the DNA that the tissue samples are stored. I already told you about it a lot. Where? That they are preserved and stored. What place am I talking about? Because I told you only about the Department of Forensic Medicine. That during the autopsy, some some samples are collected from a person? Do you have any idea? That the tissue samples are collected and stored during life of a person. The person is alive and they collect sample and they store it. Why? Why do they do it? That they collect a tissue sample and they store it for many years. Cytological samples, yes, you are right, or just pathomorphological samples. I'm talking mainly about, for example, the person who is diagnosed because of cancer, so the tissue samples are collected from the person for diagnostic purposes, and they are stored in the Department of Pathomorphology for many years. So I told you up to 20 years. So, or even longer if they did not destroy the sample. So then sometimes stem cells, but they are stored just like. So I told you, and we had at least, I think we had several such cases. We had several such cases that the putative father died because of the cancer, and I even did such examination. The man died because of stomach cancer. I got this stomach cancer tissue preserved at the Department of the Pathomorphology, so they sent us the tissue sample, and I had the mother and the child, and I compared the profiles and the profiles, so I proved that this man, who died later, because he was diagnosed when he was alive, who died later, and I showed that this man was the father of the child, okay? So the biopsy, you are right. Biopsy just for the pathomorphological exam, for the pathomorphological examination, okay? So let us go further. Great. Okay, so let's go. Let's continue. Okay, this slide just to tell you that the stability of DNA may be different in different conditions, okay? And generally, so I think it's quite obvious to you, and we do not have to go into details with it, because it's quite logical that, for example, when the temperature is lower, it is better for DNA. And now the second part of the slide tells you that the quality of DNA isolated from various tissues may be different, okay? And for example, high quality DNA may be obtained from skeletal muscles, whereas DNA obtained from spleen or from liver is usually of worse condition because of the processes which take part in these organs. Okay, let us go further. And here's just a brief history of forensic DNA typing. Okay, so in 1985, this man, Alec Jeffreys, described the so-called genetic fingerprints, which are obtained with the use of multi-locus probes. Okay, so this is what you can see under his hand, that he has such a profile which is unique to only one person in the world. So this is why it was called genetic fingerprint. In the same year, the first scientific paper on polymerase chain reaction was published. The co-author of this study was Karen Mallis, who was actually the person who discovered this technique, and who was given a Nobel Prize for this discovery 8 years later. Yeah, so this is this guy. In 1988, it was started to perform DNA typing in forensic casework. It was done for the first time by the FBI in the US. Okay, and in 1995, the first database of DNA profile was established. It was this one, which was in the United Kingdom, that I was talking to you about. That this database violates human rights. Okay, so let's go further. So here are steps of DNA sample processing shown, how we process the samples. Okay, then we get the sample. For the first step is always to get a sample from a crime scene or from paternity. For paternity testing. Okay, we extract DNA. We have to quantify DNA. And then we amplify many STR markers, which are separated by electrophoresis, and we determine the genotypes of the individuals, and we compare the profiles and compute the evidence value. Where we compute probability of paternity, or we generate a probability of random match just as a parameter which shows that this profile is unique, and it's impossible, generally statistically impossible, that another person will share the profile with the suspect and the sample found in the crime scene. Okay, so the first technique, which was developed for what do you think of 5 minutes break? Okay.
Yes.
We have still, okay, so let's have 5 minutes break because we have still many hours of our classes today. Okay, so okay, can you hear me?
Yes.
Okay, that's good. So the first method, which was developed for forensic DNA typing, was a restriction fragment, was analysis of the restriction fragment length polymorphism, RFLP analysis. Okay, so generally, you know, and I guess you have done it many times in during your studies, the procedure of DNA isolation, and which always consists of several stages. The first is always to perform cell lysis with the use of some detergents or enzymes which digest proteins and cell walls. Then we have to denature and inhibit enzymes, especially nucleases, by detergents or high temperature. Then we have to remove proteins, other impurities, and then we have to concentrate the condensed DNA and remove salts with the use of usually by precipitation of usually by precipitation of the alcohol. Okay, this may be ethanol or isopropanol. So we have sometimes if we have a lot of DNA. I remember we used to, for example, in the past I used half a milliliter blood samples for DNA extraction. So there was a lot of DNA in this volume of blood. So when I was adding ethanol or isopropanol, I could see even pure DNA just in the test tube, or it was just like a small fragment flowing in the liquid, so I can see, wow, this is just pure DNA inside, which was precipitated by ethanol. Okay, okay. So the next step is always to, after the DNA extracted, is to assess DNA quality for RFLP analysis. Okay, it is done by electrophoresis in the agarose gel. DNA may be stained with ethylium bromide or any other intercalating dye. So here you can see DNA of good quality, and here you can see something like like smears of DNA was degraded in this case. So extraction of DNA should be repeated. Okay, and another step is always to determine concentration of DNA, because we always have to use exactly the same DNA, the amount of DNA for any other steps. The first method, which is just historically the first method which was used, and it is still used by us even now, especially of good quality DNA, is the measurement of DNA concentration by spectrophotometry at 260 nanometers. Because at this wavelength, DNA shows a maximum of absorption, okay? So we can, additionally, with the use of this technique, we can assess the presence of other impurities, especially proteins which have absorption, which show absorption peak, absorbance peak at 280 nanometers. Okay, so this is just the first method which can measure the DNA concentration in case that this sample is pure, yeah, doesn't have any confounding impurities, okay? But sometimes the sample may show some other substances which also absorb DNA at which also absorb light at 260 nanometers, okay? So in this case, spectrophotometry is not good because our measurement will be overestimated. Okay, because actually, in fact, in our sample, there is much less DNA, and what we measured by spectrophotometry was a result of presence of impurities. Okay, so this is why we use, sometimes it's recommended to use fluorimetry. Fluorimetry, which uses DNA intercalating dyes. So these dyes intercalate DNA, and they modify the physical-chemical properties. So they start, for example, shining after the how do you say, excitation, after the excitation by light. Okay, so they start shining. Okay, so we measure this fluorescence of this dye. Okay, so this is one of the way which allows us to measure very precisely amount of DNA in the sample, concentration of the DNA in the sample. Okay, so what we have to prepare always in case of fluorimetry is we have to prepare the curve, yeah, the standard curve, so that we can with certain known concentrations of DNA, so that we can apply this curve to compute concentration of DNA in our unknown samples. Okay, in case of spectrophotometry, we do not have to prepare this curve because we know that this amount of absorbance corresponds to this concentration of DNA. Okay, but sometimes it's not sufficient to know the exact amount of DNA in the sample, the exact concentration of DNA on this, in a sample. Sometimes it's more important to know something else. And it's achieved with the use of real-time PCR. And then now there is a question to you. Why real-time PCR may be better sometimes in forensic genetics to measure concentration of DNA in comparison to fluorimetry, which measures very precisely the amount of DNA? Why real-time PCR may be better? Do you have any idea? Especially to forensic genetics, yeah, because okay, I know in my sample, I will tell you an example. Okay, I measured concentration of the DNA with fluorimetry, and I know that the concentration of DNA is 5 nanograms per microliter. Quite good concentration. Okay, I dilute the sample, I add to the PCR this certain amount of DNA, and I get almost nothing, very small peaks. So it means that the concentration in the sample was much lower than 5 nanograms. So what's going on? Okay, the fluorimetry measurement told me 5 nanograms per micrometer. I used this concentration to set up PCR, and the result shows that, oh no, there is almost nothing in the sample. Why did you dilute it? Yeah, so what's the problem? What was the problem with the sample? What do you think? And then I take again the same sample and I measure it with the real-time PCR, and the concentration is, let's say, 0.5 nanogram per microliter. No. It's let's assume that it specifically measured total DNA in the sample, and we are sure that this result is correct. But anyway, I use this result for PCR setup, and I got almost nothing. And I measured this concentration with real-time PCR. Oh, and actually there is much less DNA important for me as a forensic geneticist. Oh, there is some information. Contamination of with other DNA. No, contamination of, but what kind of contamination? This is a question to Carola, because you said, sorry, you said contamination of other, but what kind of DNA, what kind of other DNA are you thinking about? This is a question to Carola M. And Victor, you are talking about another explanation that you, sorry, could you? Degraded. No, okay. Maybe it is degraded, but we measured, yes. Yeah, this may be actually the reason that we, but the real-time PCR also measures us the amount of the concentration of a small fragment of DNA. So with the degradation in this measurement by real-time PCR is just because it's just it doesn't have, how to say, the degradation process has similar impact on the fluorimetry and real-time PCR measurement. Okay, of course we can design a way that the longer fragments are amplified by real-time PCR. So in this reaction we can also assess the quality of DNA, whether it is strongly degraded, because we will amplify short fragment, which will be amplified even in case of degraded DNA, and longer fragment, which will be degraded only, amplified only if the DNA is not degraded. Yeah, so there are some ways of of assessing degradation process in the DNA sample. Yeah, so this is, but generally no, this is not the good question. But there is a good question I can see. Fluorimetry isn't specific. Carola M, you are right, but what do you mean? Fluorimetry isn't specific. So so why real-time PCR increases specificity for forensic genetics? What do you mean? You are right, fluorimetry isn't specific, but specific for what? I told you it measures specifically. No, not background noise. Maybe background, no. Fluorimetry measures specifically DNA concentration. So we are sure that the concentration of the DNA is 5 nanograms per microliter. I am sure that it is the correct result. Okay, I didn't make any mistake, so I am sure. But Carola told us it isn't specific for forensic genetics. Why? Because I know the concentration of total DNA in a sample, and for me as a forensic geneticist, it is not the result I am interested in. Total DNA in the sample, And I do not want to know the concentration of total DNA in the sample sometimes. I'm only interested in concentration of some DNA in the sample, not total. Hmm? I'm not interested in the concentration of total DNA in the sample. I'm only interested in the concentration of some DNA in the sample. What kind of DNA are you interested in? Okay, because I have, I don't know, I got to, yeah, I told you, yeah, I got a blood sample from the autopsy room. The blood was decomposed, was stinking, yeah, and just a lot of gases were produced. Was so really advanced degradation processes took place in this blood sample. And I extract DNA. I measure it with fluorimetry, and it's 5 nanograms per microliter. And I measure it with the real-time PCR, and it's, I don't know, even lower, 0.1 nanogram per microliter, very low. What? What did I measure in case of fluorimetry? Yes, perfect. Yeah, finally. Okay. So this is actually, yeah, David, you are right. Also, micro, exactly. So actually, what I am interested in as a forensic geneticist is the human DNA in the sample. Yeah. So okay, the fluorimetry measured me the total DNA concentration. Yeah. And I would like to know, I would like to determine the DNA profile of a human, not bacterial. Yeah, and the PCR real-time PCR technique is specific to human DNA. Yeah, the primers and the probe were designed in a way that they amplify human DNA. Yeah, so actually the total DNA is something which I'm not interested in. So in case of such samples which were degraded and there is some DNA of some microorganisms or some other maybe species or whatever, so for me it will be interesting to get to know the total, so the concentration of the human DNA. So moreover, I can also measure with the real-time PCR the concentration of human male DNA, because sometimes we have a mixture of a DNA of a woman and a man, especially in rape cases, yeah? And I would like to determine, for example, Y chromosomal profile of the male in the sample, yeah? So I would like to know how much male DNA, human male DNA is in the sample, not just the human total DNA, okay? So this is just okay, let us go further. Okay, so let's skip it, it's not so important. Okay, and why do humans show RFLP? Why do humans show restriction fragment length polymorphisms? First of all, because humans show polymorphisms of restriction sites. That some individuals in a certain locus may have a sequence which will be digested by a restriction enzyme, and some individuals, because of some variation in the nucleotide position, will have this restriction site removed. So after the digestion, one longer fragment will be produced. Okay, so this is the the first reason. And the second reason why humans show RFLP is the polymorphism of the variable number of tandem repeats, the VNTR polymorphism. Okay, so it means that between exactly the same restriction sites, just like here, there's different number of tandem repeats. If there are less number of tandem repeats, the fragment after the digestion will be shorter. If there are more tandem repeats, the fragment after digestion will be longer. Okay, so there are two reasons why humans show RFLP. Okay, so there are, you can see different restriction enzymes which are used in forensic, which are useful for forensic genetic purposes. Okay, of course you do not have to remember these enzymes; these are just examples which are of the enzymes. Okay, so after the digestion, the products are separated by agarose gel electrophoresis, yeah, according to the size. So if the fragment is smaller, it gets quite easily through the gel. If the fragment is larger, it goes much more with more difficulty through the gel, so it goes more slowly. So this is how they are separated. It takes us several days, 3 years, even 3 days. And after the separation by size, the DNA fragments are transferred by so-called solvent blotting method from gel to the nitrocellulose filter or nylon membrane, and there are different methods of this transfer. And after they are bound to this membrane and hybridized with a probe, so which detects our DNA. Okay, so there are different techniques of labeling these molecular probes. There are techniques that are isotopic, which we have never used these techniques, luckily. So they are, of course, they are a bit dangerous for health and for the environment. So generally, currently, non-isotopic methods have developed so well that actually the sensitivity is comparable to the isotopic methods. Okay, and okay, so we get many fragments. We get many fragments of as a result of our examination, which we can compare. Okay, and we can compute so-called similarity index, which is proportion of shared bands. So this is the formula for this parameter. Two times the number of shared bands between the two individuals divided by the sum of number of bands between the compared individuals. Okay, so we can express this value as a percentage, and for unrelated individuals, for this probe MZ1.3, we get a value of approximately 24%. For first degree relationship, a parent and a child, we get a value of approximately 60%. Okay, so this is, remember, this is not any probability. This is just the similarity index. Okay, and based on this value, we can conclude that whether the person is a father or not. Okay, and moreover, random match probability, which is the probability that two profiles of unrelated individuals will be identical, is just 10 to the power of minus 12. Okay, so actually there are not so many people in the world, so it's actually impossible that two randomly selected people will have identical DNA profiles obtained with the use of this multi-locus probes. So this is why they are called genetic fingerprints. Okay, they are called genetic fingerprints, so that all people are identical. Okay, so this is just the power of discrimination. Power of discrimination is the probability that the two genotypes are selected randomly from the population will be identical. So you can see that for this multi-locus molecular probes, these profiles are unique and found only once in a population. Okay, so this is just a brief summary how the examination is performed. We have DNA, we have a biological material, we get we extract DNA, we cut DNA into pieces, we separate the fragments, resulting fragments by size. By electrophoresis, we transfer fragments from gel to the membrane. We incubate the membrane in a solution in a solution of a molecular probe, and then we detect the fragments and compare the resulting profiles. Okay, so how do we interpret these results of multi-locus analysis? Paternity is confirmed if all non-maternal restriction fragments possessed by a child are present in a putative father, where there are a lot of shared bands between a child and a putative father. What does it mean, a lot of shared bands? It means this is this similarity index. Yeah, you remember, we have to compute this value. So for example, it may be for one of the probes which I told you about, this MZ1.3, it will be 60%. So it means a lot of shared bands. Okay, and in general, we can say that relationship of restriction fragments is tested between a child and its mother and a child and its putative father. Approximately 50% of shared bands indicates parenthood, less than 40% of shared bands indicates no relationship. And paternity is excluded if a child possesses foreign bands, which are present neither in a mother nor a putative father, with a few or no shared bands between a child and a putative father. Okay, so generally, if the child possesses, but if a child possesses only one band, which is foreign, and the rest matches, does it mean that this man is not the father? What do you think? Mm-hmm.
It doesn't exclude paternity as long as the similarity in the pattern is more than 50%, as you mentioned before.
Yes, yes. But generally, okay, we have found that the child has like, okay, we have a mother and child and putative father, and we compare them, that in all the fragments found in the child, there is a band from the mother, and the one which is not maternal matches the putative father, okay? But one of the bands which is found in the child is not found in the putative father. It's not from the mother, and it's not found in the putative father.
Could it be the cause of mutation?
Yes, yes, you are right. So this is a very good idea, Adrian. Yes, you are right. Yes, they are mutations. Yes, so please remember that they are mutations. So one mismatch, one inconsistency does not allow us to formulate a conclusion that somebody is not the father. Okay. So now another variety of the technique is the RFLP analysis with the use of single locus probes. Examination is the same. The only difference is that we get less bands, okay? We get maximally 2 bands because we analyze only single locus, okay? So we get maximally 2 bands or even 1 band if a person is homozygous, okay? So this is just the only difference is that it is a different result, okay? So these single locus probes are worse because they have worse power of discrimination. Look, in case of MS8 molecular probe, every 100th individual has the same profile as mine, yeah? So there will be many people identical as me if we analyze only one single locus probe, okay? So in order to reach the comparable power of discrimination, we have to test more single locus probes. Okay, so there is more work to get a profile which is unique. Yeah, but it is still possible. So here is just a general comparison of this multi-locus probes and single locus probes. I told you single locus probes are worse than multi-locus probes because the power of discrimination is worse. Okay, but this is their only disadvantage, because in general we can say that they are better. Why? Because, for example, we know exactly how these different single locus probes are linked to each other. We know that they are independently inherited, yeah? And we know that every person has 1 band from a mother and another from a father, yeah? So we can very easily, I will have some questions, some tasks for you to solve, and you will be able to solve them because you know that every person has a band quite different. They will look quite difficult for you. They will look like exercises to you, yeah? And you will be able to solve these questions because you know that every person has a band always from a mother and always from a father, okay? Moreover, another advantage is that it has higher sensitivity. Sometimes from the crime scene, we do not have so much DNA to perform this multi-locus analysis, okay? But look, in case of single locus probes, we need even 10 times less DNA. Moreover, we can also detect DNA mixtures of several individuals. In case of multi-locus probes, DNA of one person, many bands, yeah? In case of 2 individuals mixed, many bands. Are there 2 individuals? How can I be sure? Okay, but in case of single locus probes, 1 person, 2 bands, 2 individuals, the most probably 4 bands, yeah? So this is possible to detect mixtures, okay? At least 2 individuals are mixed in case of this DNA, okay? However, this RFLP analysis is quite laborious and time-consuming, yeah? It lasts even 10 days and comprises many stages, yes? So actually, and according to the poll, and it still requires a lot of DNA, yeah? Even 50 nanograms of DNA is still a lot, yeah? So I told you, and according to the Polish law, we can arrest a person without the verdict of the court for maximally 48 hours. And if we would have to prove, if we had to prove that somebody committed a crime in less than 48 hours, with RFLP analysis, it would be impossible. Okay. So however, it's possible with the use of polymerase chain reaction, PCR. Okay. The technique is generally very similar. The technology is very similar to the previous analysis. The difference is the enzyme, okay? Because in case of RFLP analysis, we used enzymes which cut DNA into pieces, yeah? And the resulting fragments have different lengths. In case of PCR, in forensic PCR, yeah, we use an enzyme which is DNA polymerase. which amplifies fragments. And the resulting fragments have also different length. So we compare the length of these fragments between resulting fragments between different individuals. So this is how it's actually what is the most commonly performed in forensic genetics, okay? So as I told you, we amplify by PCR both mini satellites and microsatellites. However, mini satellites are currently not amplified, are not used in forensic practice because they have longer alleles, yeah, they are not suitable for analysis of degraded DNA. Okay, so this is the main problem of them. So some examples of these mini satellites and short tandem repeats. You can see that the vast majority of forensic STR markers have tetranucleotide repeats. Okay, so the repetitive unit has 4 nucleotides. I will tell you later why, but let us go further. So just the briefs, just how we'll quickly remind you how the PCR works. Yeah, so we have double-stranded DNA, which is denatured, primers bind, and the polymerase elongates. So if we have one molecule at the beginning, after another cycle of DNA, of PCR, we get 2 molecules. Then we get 4 molecules, then we get another cycle, 8 molecules. And assuming 100% efficiency, after 32 cycles, we get more than 1 billion or milliard. Yeah, milliard is a Polish or Spanish word, to billion, British billion, British English billion, because it's old-fashioned actually, so it's not used anymore in the British English. Because the English language has a lot of mess with the naming long numerals. So generally, we get more than 1 billion copies of targeted DNA, assuming 100% efficiency. So it's perfect tool for forensic genetics, because very often we encounter minute amounts of DNA. Yeah, so we apply this PCR, and from this minute amounts of DNA, even 1 cell may be sufficient to generate a DNA profile, which will tell me who this DNA came from. Okay, so however, this extremely high sensitivity, which is a very big advantage for forensic genetics, is also a problem. Why? My question to you. Why is extremely high sensitivity a problem for forensic genetics? Let me read. Contamination. Yes, you're right, Carmen. Okay. Yeah, there is actually, this is the main problem, is the extremely high risk of contamination. Yeah, we have to be really very careful when we extract DNA. When we collect a sample, yeah, from a crime scene, yeah, when we extract DNA, when we set up PCR, because if I do not work sterilely, it will show up that I committed, and I sneezed there, then it will appear that I commit all the crimes in the area, yeah, or it will not show up that I'm the father of all the children in the neighborhood, because it's not possible. But this is just the problem, yeah, contamination, and then there were some cases of contamination which were really dangerous, yeah, and they were announced even to the public, yeah, to the media. I remember even a case, there was several years ago, there was a case of a man who was committed of a crime in Manchester in the UK, actually in a city that he has never visited in his life. And they found that the reason why his profile was found in Manchester was actually contamination in laboratory. Yes, so this is just the problem that you have to be encountered. So this is why we use very different procedures to prevent this contamination. I told you about this sodium hypochlorite, UV light here, the gloves, the laminar hoods, and so on. So we protect the samples. Actually, the most important thing is not just to protect us from the sample, that we will get, I don't know, infected by the sample. No, the main task in forensic genetics is not to protect the technician, but to protect the sample, yeah, from contamination, actually. Okay, let us go further. Okay, so the allele, so when we amplify the short tandem repeats, they have different, I told you they have different repetitions, different number of repetitive units. If there is, for example, here is an example, we have primer pairs. So here, yes, so they amplify allele with 7 repeats. It's called, in forensic genetics, it's called allele 7. So it's sequenced, and we know that this length corresponds to 7 repeats, and it's called 7. And there is another allele with 8 repeats, okay? So this is how the allele calls alleles are called in forensic genetics. Okay. So and the variety of PCR which is used the most commonly in forensic genetics in the world is so-called multiplex PCR, in which many markers are amplified simultaneously in one reaction. So we use many primer pairs to amplify many markers simultaneously. So thanks to this variety, we get much more forensic information per time unit, okay? So we have higher power of discrimination. We have to work less, yeah? The labor is reduced to obtain results. So it is good for us as technicians, so we do not have to work so much to get the result. And it's also good for our clients who are courts, prosecutors, police, yeah? Because if we work less, the final cost will be lower, yeah? So this is also good for them, that the cost will be lower, so they will have to pay less for the examination, okay? Also, less DNA is required to get a unique profile, okay? Because if we, for example, I don't know, have a DNA from one hair bulb, okay? So I don't know, I can set up 2 PCRs, maybe 3 PCRs from this amount of DNA, yeah? And if I analyze marker by marker, I will not get a unique DNA profile. There will be many people identical. But if I amplify and know 15 markers in one reaction, the profile will be unique, yeah? And I'm sure that this is the person this is the same person, okay? And also the risk of contamination is reduced because we do not have to open our test tube so many times. We just open it once and get so this is another advantage, okay? And here you can see examples of the first multiplex systems that were developed for forensic purposes. They were separated by electrophoresis in polyacrylamide gels and stained with silver. So they are called triplex systems because 3 markers were amplified in one reaction. Okay, so the markers were identified by the size of the product. Okay, the shortest fragments belong to THO1 marker, whereas the longest fragments belong to CSF1PO marker. Okay, so you can see, so I will show you how they look like. Oh, here is an example of how they look like, okay? So this is a mixture of alleles found in the population, observed in the population. They were sequenced, and we know exactly that this allele is called 9 because there are 9 repetitions of the unit, okay? This allele is 11 because there are 11 repetitions of a unit, okay? So this mixture of alleles is called allelic ladder, okay? So you should know what the allelic ladder is. So this is the mixture of the alleles, okay? So, and you can see results of amplification of DNA of different humans, yeah, of different individuals, okay? So by comparison with the ladder, we know that this person has genotype 17, 19, 15 in case of this marker, 15, 18 in case of this marker, and so on. So this is how. So this allelic ladder is used also to determine genotype of the tested individual. Okay, and until now, and also in forensic genetics, allelic ladders are used. I will show you how they look like later. Okay, so we separate these fragments by capillary electrophoresis. Okay. Maybe let's have a short break. What do you think? 5 minutes for the last time, okay? Because there is 5 hours, so many Okay, what do you think? 5 or 10, maybe 10 minutes, 10 minutes. Okay, longer. Let's have a longer break. What do you? Is it okay for you?
Yes.
Okay, thanks a lot. Bear with me. Yes. Yes, are you here?
Yes.
Okay, let's start. Mm. Share screen. Okay, a technique which is used for routinely in forensic practice in forensic laboratories around the world is capillary electrophoresis, which separates the PCR products of different length by size. So there is a what we use is a very thin capillary of a diameter of 50 micrometers, and there is a fragment of this capillary which is called window, onto which ion laser shines, and argon ion laser shines, and it uses fluorescence in fluorescent dyes which are used for labeling resulting PCR proteins. Okay, so after application of an extremely high voltage, usually 15 kilovolts. Okay, so our DNA starts traveling through the capillary from minus to plus. Okay, and it goes. Okay, the shorter fragments goes quicker. The longer fragments go more slowly. Yeah, so they go, migrate until they reach this window when and the fluorescence is induced and collected by the detection system, which is stored in the form of pics on the computer, and we compare these profiles in form of pics, not of pens, but in form of pics. Okay. So here is a 310 genetic analyzer, which was actually historically the first capillary electrophoresis unit developed for forensic purposes. Okay, so actually we have such a unit, such an apparatus, such an instrument in our department. So actually it's not used anymore in our practice, but we still have it. So actually I use it only for students. When I have some laboratory classes, I open this instrument to them to explain how everything works inside this instrument. Okay. I'm sorry. Okay. Okay, so here you can see zoom at the inner part of this instrument. So here is our capillary that I was talking about to you. So this is our capillary. Here you can see the end of the capillary with the electrode, platinum electrode. Here is an autosampler with the tubes containing our samples, which are our PCR products used for separation. Okay. So here is so this is one electrode, here is another electrode. So this is the voltage is applied between this electrode and this electrode. This high voltage of 15 kilovolts is applied. So DNA goes from this vial, goes up, goes. So this block is heated. So there is 60 degrees Celsius is used for separation of DNA fragments, as in forensic genetics. Or it can be also used for sequencing, so that the temperature used in the block is 50 degrees Celsius. Okay, so DNA is denatured, so it goes in a single-stranded form, goes until it reaches this window, and there is laser shining onto this window all constantly, so that there is the resulting fluorescence is collected by the detection system, so detected by the collection system and collected and stored on the computer. Okay, so this is how the and here you can see just the close-up of the sample injection site. Here is the capillary and the electrode, so they enter together the test tube for collecting the sample. Okay, so so here you can see how the resulting fragments travel through the capillary. Yeah, so this is the laser ion. The argon ion laser shines onto this window. This is the detection site. The fluorescence is produced, and it's collected by this CCD camera, and it's, I told you, and it's stored on the computer. Okay, and what you would have to I would like you to understand that in case of capillary electrophoresis, we do not inject any volume onto this instrument. I guess you have done maybe in your laboratory work, for example, high performance liquid chromatography, yeah, HPLC. And in case of this technique, we use a special amount of liquid, and we inject this volume onto the instrument. In case of capillary electrophoresis, we do not inject any volume. What we do is electrokinetic injection. So actually, we put the capillary and electrode into the tube, okay, containing DNA, and then extremely high voltage is applied so that the DNA goes up to the capillary. Okay, and moreover, it's very important that we have to use for suspension of DNA, for suspension of our PCR product, we use deionized formamide or deionized water. Do you have any idea why the formamide must be deionized for capillary for electrokinetic injection in capillary electrophoresis? Do you have an idea why the formamide, eventually water, yeah, alternatively water, must be deionized? Charge, yes, charge. But what would happen if the formamide were not, if the formamide was not deionized? Pardon? No. It's very important that there are no ions in the formamide that we use, no other ions. Because if there are more ions, then what would happen? We put the capillary and the electrode inside. But I will mark. They will block the capillary. Yes, interfere. Yes, what do you? Yes. You are right. Okay, Victor, you are right. I agree. But how will they interfere? They will actually they will interfere in a way that they will compete with DNA. Yeah. So we apply the voltage, and instead of DNA, instead of DNA, only these ions, small ions, will be collected. Yeah, will be injected. Yeah, electrokinetically to the capillary. Okay, so the signal strength of our PCR products will be very, very low, because instead of DNA, we injected mainly some ions from the formamide or some ions from water. Okay, so this is why it is important to use a deionized formamide in capillary electrophoresis. Also, prior to electrophoresis, DNA must be denatured and immediately cooled down, and so that the DNA is separated in the single-stranded form. Okay, let us go further. As I told you, whether we use that the capillary is heated to 60 degrees Celsius in case of analysis of DNA fragments, as in case of forensic genetics. Okay, and also the urea is present in the buffer, which prevents renaturation of DNA and forming of secondary structures. Okay, so just the high temperature and the urea are factors which prevent renaturation of DNA and the forming of secondary structures. Okay, so so I told you that the DNA goes through the capillary. It interacts with the cross-linked polymer, which is used for filling the capillary. Okay, so prior to each electrophoresis, the polymer is exchanged. So new portion of the polymer is pumped to the capillary. So the old one is removed, and then the DNA goes to this polymer, and the longer it is, the longer DNA fragment is, the more interacts with this cross-linked polymer, and so the more slowly it goes through the capillary. Okay, and there are different dyes used for labeling PCR product. And here you can see just a set of four dyes which are used for labeling. So they have, you can see different emission spectra of these 4 dyes. Okay, you can see that they have different peaks. Yeah, that fluorescence maxima of this dyes are totally different. Yeah, but we never. But come on. For example, when I measure light at this wavelength, okay, 580 nanometers. Of course, the yellow dye will shine. But also, if there was green dye, that it also shines. Yeah. If there is also blue dye, it also shines. And also there is a small fluorescence emission also for the red dye. Yeah. So this is why we have to use something which is called mathematical matrix. Okay. So there's some visual filters applied, and some mathematical matrices which removes some fluorescence of some other dyes. For example, if I measure fluorescence in this window, okay, and there is something shining in this window, it means that we have to extract this light in order to make this peak totally different. Yeah, it's quite difficult to explain to you, but thanks to this, we can see only pure colors, okay? Because it's not only green color. There are many colors in case of these dyes, yeah? But the matrix makes this color only green, okay? So this is how it works, okay? Let us go further. And some other filters used in forensic genetics. Okay, so you can see here an example of a kit which is called Profiler Plus. It amplifies 9 STR markers and a melogeninous agenda marker. Okay, and so we can differentiate this marker from this marker and from this marker by size, because they have different sizes, yes, size ranges. Okay, but we can differentiate this one from D3, from D8, and from D5 by color, yeah, because different dyes were used for labeling 5' ends of primers, and also the resulting PCR products have also different dyes at the 5' ends. Okay, so here is an example of an of amplification of the identifier kit, which became one of the most commonly used kits in forensic genetics. So 4 dyes were used for labeling PCR products, and the 5th dye is used for labeling size standard. Okay, which is used for computation of the sizes of the resulting products. Okay, so we use the standard to create the standard curve, and this standard curve is used to compute the sizes of this fragment. And when we know the sizes of this fragment, and if we have the allelic ladder, we can compare the sizes to the sizes in the allelic ladder, and we know that this genotype of this person in the FGA marker is 21, 22, because the same sizes were obtained for the alleles 21 and 22 in the allelic ladder, just the standard mixture of the alleles. Okay, it's an example of paternity testing here. This is the father, the mother, and three kids. Okay, you can see that the child has always something in common from the mother. These are these red alleles, and from the father, these are these blue alleles. Okay, so this is not something which let's skip. It is not important. Okay. A very important part of forensic genetics concerns population studies. Why? Because first of all, one of the reasons is that we have to find all the alleles or majority of the alleles observed in the population. We have to sequence them, we have to mix them, and we have to create this allelic ladder. Okay, so you know what this allelic ladder is. Okay, during this year there will be some exercises for you also, so you will see such ladders also on your computers. So you are going to work in groups, and you will see how such results really look like. Okay, but not today. And the other reason is that we have to that our expertise, our opinion, our is preceded by statistical evaluation. We compute evidence value that this suspect is a source of DNA in the crime scene. Okay, we compute probability of paternity, that this man and no other man is the father of this child. And for these computations, we need allelic frequencies. We need to know what the frequency of the allele is in the population. Okay, because for example, if a child inherits allele which is very rare in Poland, then probability that this man is the father of the child will increase a lot. Okay, it will be very high. But if this allele is quite common in Poland, yeah, the probability will increase only a little, or it can even drop down, because every second man in Poland has this allele. So why do you think that I am the father? Because if there are so many men who have this allele, yeah, so this is why this probability can even drop down for such a very common, very frequent allele in the population. Okay, so this is another. So this is why another reason why we have to know the frequencies of the alleles in the population, and also we have to assess usefulness of a marker in forensic genetics, because sometimes some markers are not very useful. So it's not worth to spend money on examination of this marker if it's totally, I don't know, there are only 2 alleles you found in Poland, and with a distribution, I don't know, 90 to 10%, so 90 to 10%, 90% and 10%. So actually, no, it will be not very informative for paternity testing. Okay, so there are some parameters shown here, but I will tell you later what it means during the part concerning statistical analysis in forensic genetics. Okay, so here is, you can see how the allelic ladders are formed. Okay, and something quite important word. Okay, microvariants. Okay, please remember this word because there are at the final exam there is a part of exam which checks your knowledge, whether you can use some specific forensic genetic terms, whether you understand what they mean. So microvariant alleles are alleles which have incomplete repetitions. Okay, so here is an allelic ladder for a marker D3S 1358. So this is the allelic ladder. This is a mixture. So we know that this person is heterozygous, has 2 alleles. Okay, the first one matches 15, so it's like a regular normal allele, 15. And the other one falls in between 2 alleles, okay? So it's called microvariant or intermediate allele, okay? Microvariant alleles have incomplete repetitions. So we know that allele 15 means that there are 15 repetitions of a unit, 15. And this allele is called 15.2 because there are 15 units and 2 more nucleotides, okay? There is an incomplete repetitive unit, okay, incomplete. So it's possible that the allele will be called 15.1, 15.3, sorry, 15.1, 15.2, 15.3, and 15.4 is impossible because it will become already allele 16, okay? Because 4 nucleotides means a complete repetition, okay? So this is, so because this marker D3S and so on, and has tetranucleotide repeats. Okay, so this is what these microvariants are. Okay, and then here you can see the reason why majority of forensic genetic markers have tetranucleotide repeats. The main reason is the production of starters during polymerase chain reaction, during PCR. Starters are artifacts which are artifacts which are produced during polymerase chain reaction, during amplification by PCR of short tandem repeats. They are usually shorter by 1 unit from the correct allele, okay? And if the allele is short, sorry, if the unit is shorter, the more artifacts are produced, okay? So here we can see example of the mononucleotide repeat. You can, and here we can see a result in case of, in the form of bands, yeah, and peaks. Very many stutters. I even do not know what the correct genotype of this person is. Okay, mononucleotide repeat. Okay, you can see dinucleotide repeats. Okay, this is this and these are correct 2 peaks. Okay, but you can see 30% of the product is the stutter, then 44% in the height of the second allele is the stutter, and there is even a stutter of a stutter. You can see here. Yeah. So there's, it's also possible, you can see also the stutters on the gel, yeah? So this is the stutter, and what is this? This is also, I guess, a stutter. So yeah, this is a stutter, yeah? So this is, it's quite difficult. In case of this marker, it's totally impossible to analyze it from the forensic point of view. In case of this, okay, if I have DNA of 1 person, it's okay, I will decipher this result. But if I have a mixture of 2 people, I don't know, is it another allele, or is it a stutter? Oh, it's quite complex. Whereas in case of tetranucleotide repeats, the stutters are quite low, approximately 10% of low or less. This is quite short. You can see here, yeah, this is the shadow. These are their stutters. Yeah, so they are quite low. So if we see something which will be, I don't know, 20% high or 25% high, I say no, it's not a stutter. This is an allele of another person. Yeah. This is a minority complement in the mixture, okay? And even there are longer repetitive units like pentanucleotide repeat markers. The stutters are even lower. You can see less than 1%. So they are perfect actually for analysis of mixtures. But the problem is that pentanucleotide repeats are not so popular because they have larger size ranges. So this is the first reason, and also they are. So this is so they are not very good for analysis of degraded DNA because of the larger DNA products. Okay, so let us go further. And here you can see examples of different allele frequencies observed in the 3 ethnic populations of the United States of America, of mainly European origin, of mainly Sub-Saharan African origin, and of largely Native American and quite admixed, and also admixed origin. They are called Hispanics. Okay, so you can see that the allele frequencies can be totally different in these different populations. It can be totally different. Okay, and now my question to you is, okay, let's imagine such a story in which we have a Polish woman who accuses of paternity a Brazilian man. Okay, and let's assume that the allele frequencies in Poland and in Brazil are totally different. Okay, let's assume. So my question to you is, which allele frequency database should be used for computation of probability of paternity in this case? The Polish one or the Brazilian one? And why? Let me read. Brazilian. But why Brazilian?
Because we're checking is he a father, and it will now make sense if we check in if mother is a mother.
No, I'm not convinced with this explanation. I will tell you. I will help you. Sometimes it's justified to use the Polish database, and sometimes it's justified to use the Brazilian database. When? Okay, let's check. Let's assume that the man matches the child. I can see, the man matches the child, so I am computing the probability of paternity. I use the Polish database, and for the Polish database is 99.99999%. Okay, 6 nines in total. So I can write in the conclusion that this man is the father of the child. But then I computed the same parameter, probability of paternity, for the Brazilian population. And it's only 99%. So it's not so high. It's not sufficient to write that he is the father, according to some recommendations, yeah? So which value should I report and why? Because one of them allows me to write that he's the father. And the other one, for the Brazilian population, doesn't allow us to write that he's the father. To write that he's the father.
Don't we need to check first if the mother were traveling to Brazil or so, to check if some other people might be the father?
Yes, perfect. Yeah, and so if she traveled to Brazil, which database should be used?
Oh, which one?
If she traveled to Brazil?
So Brazil. So
No, yeah,
Brazil.
exactly, Brazilian one. So actually, the idea is to check whether he is the father of the child. So we compute whether he is the father of the child or any other person from the surrounding population. Okay. So if they met in Poland, who could be the father of the child? Either the Brazilian who was in Poland or any person from surrounding Polish population. So we take the Polish population for computation. Okay, so because we would like to know what are the frequencies of these alleles in Poland, yeah, and we see, oh, they are very rare in Poland. So actually, probability that he's the father, because this allele is so rare in Poland, the probability that he's the father is very high, yeah. But if they met in Brazil, who could be the father of the child? Either the Brazilian who she met in Brazil, or some other Brazilians, yeah. And maybe these alleles found in the child are very common in Brazil. And they do not support strongly that this Brazilian is the father, and we would have to test more markers in order to prove that this man is the father of the child. Okay, let us go. Let's continue. Okay, so here you can see the different values of power of discrimination and exclusion chance for different markers using DNA markers using forensic genetics. Yeah, you can see that these values, you remember the values for in case of serological markers, they were much worse. Yeah, really much worse because of the number of alleles which are because the DNA markers have many more alleles than serological markers. Okay, and in case of this marker D1S18, we have 64% chance that the man who is not the father will be excluded of paternity. So more than half of non-fathers will be shown that they are not fathers. Yeah, so this is a very good. But we never test only one marker. We always test many markers. So of course the exclusion chance grows very rapidly the more markers we analyze. Okay. Okay. Now I'm going to tell you a bit about the Y chromosome in forensic medicine. The Y chromosome is actually one of the smallest human chromosomes. Only chromosomes 21 and 22 are shorter than the Y chromosome. And what is important about the Y chromosome that 95% of the chromosome does not undergo recombination during meiosis. Okay, only these yellow labeled fragments called pseudo-autosomal regions recombine with the X chromosome during meiosis. Okay, whereas the rest is actually the largest haplotype in the whole human genome. Okay, haplotype which is transferred always as one fragment from generation to generation. Okay, and it has some special features which makes it a special tool in forensic cases. First of all, it's specific only to males. Okay, and I don't remember, but I have read once that I think 93%. I have read some statistics about I think England and Wales that 93% of crimes committed with the use of force are committed by males. Okay, 99% of crimes with sexual background are committed by males. Okay, so there is a quite common situation in forensic casework that an offender is a male and a victim is a female. Okay, and there is and that we have mixture of DNA of a man and a woman. okay? And if we analyze autosomal markers in such a mixture, then if we are lucky, and if there is a lot of male DNA, then we will see a mixture, yeah? We'll see that they are like I don't know, the ratio is like 1:1 or 1:2. You will see a mixture. Then we'll see alleles, we will detect alleles of another person. But if the ratio is, I don't know, 1:100, yeah, there is only small amount of male DNA in the mixture. Like we have a vaginal swab, there are no sperm cells at all, and there are only some epithelial cells of the offender. So we will not detect the male DNA, yeah, if we analyze autosomal markers. But if we analyze the Y chromosome, the woman does not have the Y chromosome. Okay, so the ratio does not matter. It can be even 1:100. It can be even 1:1000. The primers will find in the mixture, will find the Y chromosome, and they will amplify markers of the Y chromosome. Okay, so this is what the first special situation of special application of the Y chromosome is the detection and discrimination of male DNA in male-female DNA mixtures. Okay, male component, yeah, in this male-female DNA mixtures. Okay. Oh, moreover, if we have a gang rape, yeah, if there are more than one offenders, we can determine the minimal number of offenders of the rapist in such a case, in such cases. Okay, so this is another application. If we detect, I don't know, like 2 alleles and in some markers of the Y chromosome. Okay, and another special feature of the Y chromosome is its unique inheritance pattern. Okay. For example, which can be used in some deficiency paternity testing cases when putative father is not available in the for the examination. Let's imagine that this woman would like to determine paternity of her son, this boy, yeah, and this putative father is not available for the examination because I know he's dead, he is abroad, we do not have him. Okay, instead of this man, we could test his father, his brother, his son, if he has another son, even his paternal grandfather, paternal uncle, paternal cousin, all of them should have exactly the same Y chromosomes, okay? And I can tell you, I proved paternity several times in a way that I proved that the tested man was the paternal grandfather of the child, of the boy, yeah? Or paternal uncle of the boy. Okay, so this is just, so I didn't have the, I showed that they are related, and the Y chromosome was actually the marker which provided me the most, which was shown to be the most informative in the case. Okay, and I told you that so you know that the men inherit Y chromosome and mitochondrial DNA from the mothers, but men do not transfer mitochondrial DNA to next generations, whereas women inherit only mitochondrial DNA and they transfer this mitochondrial DNA to next generation. And also, I think I will. Also, so Y chromosomal studies also. Also support are also used in some anthropological examination about human origin, about human migrations, and actually they support the out of Africa model that all modern humans can be traced back to one Y chromosome who was possessed by the genetic Adam who lived many years ago somewhere in Africa. Okay, so actually all modern Y chromosomes can be are different from each other because of mutations. Okay, mutations which are which can be, when we reverse these mutations, we go to the most original Y chromosome which is found, which are found still in Africa. Okay, and also this, since we know that these mutations are generate so-called haplogroups. Okay, haplogroups. This is like a unique mutation which gave rise to like a branch in the Y chromosome. So all males who belong to the same haplogroup are somehow related. They shared the same male ancestor many years ago. Okay, so but these haplogroups have very uneven distribution in the world. Yeah. For example, in Europe, we have mainly haplogroup R, I, so for example, sometimes it can be useful to answer the question about the geographic origin of a culprit, okay? Because, for example, if I detect in Poland haplogroup A or B, this haplogroup of the Y chromosome, this haplogroup is found only natively in sub-Saharan Africa. And there were no migration from Africa to Poland, just, I don't know, a historical migration, yeah? Only eventually right now, nowadays, okay? So if I detect this haplogroup in Poland, I can be pretty sure that this is a person of recent sub-Saharan African origin. So I even can suggest that this is a person with black skin, okay? So this is, and this can facilitate the investigation of a case a lot, okay? So let's go further. So there are also some problems related to the application of the Y chromosome in forensic casework. This first problem is that we cannot prove a crime, that this person committed a crime only based on the Y chromosome. Because of paternal relatives share the Y chromosome with the culprit, okay? So this is the important problem, okay? Because we can imagine a man who claims, why do you think that I committed the crime only based only on the Y chromosome? Because my father and my brother are also criminals, yeah? And they also have the same Y chromosome, okay? So it's for me, so it doesn't mean that this was me, okay? And another problem is, but there are also some markers which were developed quite recently, which were discovered quite recently. They are called rapidly mutating Y chromosomal short under repeats. They mutate so much, so quickly, that if we analyze a set of 13 rapidly mutating Y STRs, then we have approximately 20% chance that the man who is not the father, sorry, that we will have 20% chance that the son will be different from a father. Okay, so I think 20% is quite a lot. Okay, so you can see that. But the problem, of course, is with the Y chromosome is the paternal relationship, and the other problem is the demographic history of a population, because some populations were established by only several males, yeah, because of some founder effects or some bottleneck effects, that population size that dropped down, and then the population size increased again. So then, as an example of such a population is the population of Buryats living at the Baikal Lake in Siberia. So they have a special social structure throughout their history, which was there was like a hierarchy, that men who were high in the hierarchy had many children, yeah. Whereas males who were low in the hierarchy, they had usually even no children, yeah. So because of this social structure, the diversity of the Y chromosome in the population of Buryats was decreasing from population to population. Okay, so because of this, you can say, oh, it's useless to test the Y chromosome if there is a crime, if there was a crime in a Buryat village, yeah, because probably most of the males in the neighborhood will have exactly the same Y chromosome. Okay, so let us go further. Now something about the mitochondrial DNA. Okay, generally, you know that the mitochondrial DNA is inherited paternally, always from the mother. It can be used sometimes to trace distant maternal relationship. Yeah? Just like, for example, you can see that this cousin and this second cousin, this great grandnephew should have exactly the same mitochondrial DNA. Okay, what is special about the mitochondrial DNA is that we do not analyze the length polymorphism. Okay, we do not amplify STRs. No, what we do is that we amplify and sequence so-called hypervariable regions, hypervariable 1 and hypervariable 2, sometimes also hypervariable 3. So we sequence, so we compare sequences of the mitochondrial DNA. Okay, we do not check the length. We compare the sequences. This is why this technique is more complex, it's more costly. So whenever possible, we try to analyze STRs, but sometimes there are situations that only mitochondrial DNA analysis can help. When we have extremely highly degraded, very old human material. This can be even, I don't know, some bones from Paleolithic, yeah, or I don't know, some very, so there is much higher success rate for examination of mitochondrial DNA than nuclear DNA. What do you think? Why? Why we are more lucky when we analyze mitochondrial DNA than and not nuclear DNA in the case of highly degraded human material, very old human material? We are slowly approaching the end of our presentation, of our meeting.
Could it be because mitochondrial DNA is circular, so it's less prone to degradation over time?
I will. It is. You are right. That it's circular. You are right. Maybe at the beginning it matters a bit. Yeah, some protects. I don't know. Let's say the exonucleases will not work on circular DNA, but after some time, the circular mitochondrial DNA also becomes linear because of some degradation processes. Yeah, so maybe at the beginning it matters a bit, but later no. But I like your idea. Yes, I like it a lot. So some other explanation. What is special about the mitochondrial DNA in comparison to nuclear DNA? That makes it more useful in analysis of highly degraded DNA. It's not the matter of the traits of mitochondrial DNA. It's not the matter of the traits of the characteristics of the mitochondrial DNA. It's the matter of what? Where is DNA, mitochondrial DNA located? Come on. In mitochondria, exactly. Yeah. There is a suggestion. Martina, I will write it. Yes. It's protected in the mitochondria. Yes, at the beginning, maybe some this mitochondrial membrane somehow protects, yeah, but there's also a nuclear membrane. But no, it's not the answer that I would like to know, to hear from you. What? It's located in mitochondria, okay? Yeah. And the nuclear DNA is located in the nucleus, yeah? And now let's imagine the cell, and let's imagine that there is a nucleus and the mitochondria. And now let's start shooting with, I don't know, with a gun. DNA becomes degraded. And now let's look at the cell again. What survived? Mitochondria, maybe. Nuclear DNA. But why mitochondria survived? No. Yes, okay. Yes, you are right. Carola, you are right. There are many mitochondria. Yes. Carola and Martina, you are right. Martina and Carola. Yes, actually, the reason is that there are many mitochondria in the cell, and there are many copies, several copies of mitochondrial DNA in each mitochondria. Yeah. So probability that mitochondrial DNA will survive degradation is higher because there's simply much more mitochondrial DNA, and there's only one nucleus in the cell. Yeah, and then actually, and there are only two copies, actually different copies of nuclear DNA in the nucleus. Okay, so they are so I like so these are the correct answers. Yeah. Let us go further. The size of the mitochondrial DNA, because Martina suggested that the size of the mitochondrial DNA matters because it's smaller. But actually, when we analyze the nuclear DNA, we also analyze very short fragments. Okay, so this is not the good suggestion, right? But I like them anyway that you think. Okay, and so another problem with the so this is the first application of the mitochondrial DNA. Another application of mitochondrial DNA is that if we have hairs without roots, it's quite common in forensics to encounter hairs without roots. Yeah, and if it's totally useless to analyze nuclear DNA in case of such hairs, because there is no nuclear DNA at all. There are only mature keratinocytes in these hairs. Okay, so they do not have nuclei. Okay, just remember, and also distant maternal relationship is also possible only to use mitochondrial DNA in case of this. In case of such distant maternal relationship, and also we can determine the haplogroups, maternal haplogroups. It's the same story as with the Y chromosome. Okay, and the main drawback of mitochondrial DNA analysis is that, of course, the same just like as in case of Y chromosome. All maternal relatives have exactly the same mitochondrial DNA. Moreover, there are quite many some sequences of the mitochondrial DNA which are very common in populations. For example, the most common sequence of the mitochondrial DNA in this non-coding region, this hypervariable regions 1 and 2, the most common sequence is found observed among 4% of Europeans. 4% of Europeans, it means that every 25th European has exactly the same mitochondrial DNA sequence, okay? So actually, come on, why do you think that it was me? If there are so many, I don't know, Europeans with the same mitochondrial DNA based on the mitochondrial DNA analysis, okay? So let's continue. Okay, so the same story with the mitochondrial DNA haplogroups, which support the evidence that all modern humans originate from genetic EVA, because the mitochondrial DNA haplogroups can be also traced back to one original haplogroup living somewhere in the Africa. Okay, so this is the comparison of the mitochondrial and nuclear DNA. So you have already told me this, that the size is, of course, of the genome is larger, of the nuclear genome is larger than the mitochondrial DNA. The majority of the nuclear genome is non-coding. In case of mitochondrial DNA, only small fragment is non-coding. Okay, this is this non-coding region which is sequenced for forensic purposes. Okay, and you know that there are 2 copies of the cell which recombine. And in the case of mitochondrial DNA, there's no recombination, and there can be even from 50 up to several thousand copies per cell in case of mitochondrial DNA. Okay, so please look at the exclusion chance of the serological markers. And look, 11 most commonly used analyzed serological markers give us 90% chance that the man who is not the father will be excluded. Of course, we can test more markers, more, a lot of work, a lot of money, a lot of another examination. But look, 10 STR markers, 10 DNA markers, amplified in one reaction, one multiplex reaction, very quick capillary electrophoresis, and we get a mean exclusion chance of more than 99.999%. We never test 10 markers, we always test more markers than 10, at least 15. But look, of course, if there is a suspicion that the tested man is only a brother of the real father, then exclusion chance is not so high, okay? You can see that for 10 STR markers, it's only 98%, but do not worry if there is a suspicion that this man can be only a brother or a father or a son of the real father, then we treat this case in a different way, and of course we test more markers, and we also assume different hypotheses for statistical analysis, and sometimes suggested to include also the brother of the offender or whatever, okay? So, and here is a brief comparison of the serology and DNA analysis. I think we will compare it, let me see, after, during the next meeting, okay? Because I think it's 11:15, so I would like you to stay with me longer, okay? So thank you very much for your meeting with me. we will see again in November. Yes? Thanks a lot. Bye-bye.
Thank you. Goodbye.
Thank you. Goodbye.